Hi, everyone. My name is Natalie Gill. I'm a bioinformatician in the Gladstone Institute's Bioinformatics Core. And today I'm going to be talking to you about ClustOpt, which is a tool we developed to optimize the clustering resolution parameter for multi-subject single-cell studies. So the motivation for us developing this tool is that we wanted to cluster single-cell data in order to identify cell types, but we wanted to avoid subject-wise batch correction, which could potentially remove biological variation of interest. And so our primary concern was balancing the influence of individual heterogeneity with our desire for granular cell type annotations. So there are other methods out there that try to address this issue of or over versus under clustering. And a lot of them suffer from double dipping, or they do not account for within subject correlation. So to account for within subject correlation of gene expression, we use a cross-validation based approach. And then to account for the double dipping problem, we use a feature splitting based approach. And so the holdout cells are projected onto the training principal components. And then that entire space is split into odd and even PCs, where the clustering and cluster evaluation are performed in the odd principal component space, and the random forest training and prediction are performed in the even principal component space. So what that looks like in terms of the algorithm is that we have our cross-validation loop, and then we repeat this for every resolution. We have our training and test split, where we perform PCA on the training subjects. We then split that space into odd and even, where the odd is used to cluster the training set with the specific resolution, and then the even is used for training the random forest model. We then use the test cells projected onto the even PC space to predict their cluster labels. And then on the odd PC space, we evaluate their silhouette scores. So if you repeat that for every subject and every resolution, what you end up with is the average silhouette score curve that looks like this, where the point at which it starts to drop off dramatically would be the most reproducible clustering resolution parameter across all of your biological replicates in your study. So just to reiterate the point about double dipping, the reason we're splitting the principal components into odd and even PCs is that we want our clustering and evaluation to happen in a space that's completely orthogonal to the training and prediction space. So to evaluate the performance of ClustOpt, we ran it on this Asian Immune Diversity Atlas dataset that has over 1 million cell types from 500 subjects, 5 ethnicities, 3 countries, and 33 cell types were annotated by the IDA authors using a supervised reference mapping approach. So ClustOpt was run on 6 random subsets of this data, where we balanced the number of subjects by sex and ethnicity. And then to evaluate how well the reproducible resolution from ClustOpt is able to find these annotations, we split the annotations into 3 levels based off their cell ontology. So the most basic cell type labels are L1, and there's only 8 of those. And then L2 has 11, L3 has 14, and each is more granular. And so we would expect ClustOpt to maybe find L2 or L1, but L3 would probably require subclustering since it is an unsupervised approach. So we calculated the adjusted rand index set for each of these labels, each of these annotations, at all of the resolutions that we tested. Here I'm only showing 4 of the subsets, but the rest of the subsets not shown follow a similar pattern. And the ClustOpt resolution here is indicated by the circle or the cross. And that's just whether you look at the average silhouette score across all cells or average silhouette score across all clusters. And we can see that for each, across all of them, there's this peak in the L2 and L3, and ClustOpt seems to be reliably finding the peak in the L2 annotation for the adjusted RAN index. And the process would be that you take that, clustering at 0.1 or 0.2, and then you evaluate whether any of those clusters have mixed cell type signatures. And then you would run clustopt just on that individual cluster to determine the subclustering resolution that's most reproducible across your biological replicates, so that you're balancing your desire for granular cell type annotations with the impact of individual heterogeneity. So just to summarize, clustopt can help guide the selection of the resolution parameter for Louvain clustering. It accounts for within subject variation in gene expression and avoids double dipping. So it can help reduce subjectivity in these unsupervised clustering-based approaches to find cell type annotations. And just like any method, it has limitations where extremely high subject-specific variation across all of your subjects could result in a flat curve, so no real reproducible resolution. And then it might perform poorly on data sets with a small number of subjects and requires a fair amount of computational resources. It is available to try if you'd like to try clustopt. Just look at the Gladstone Institute's GitHub page. And I just want to acknowledge everyone who had a part in developing this. So Ruben Thomas is my mentor for this project and helped guide the algorithm development. Minyoung Shin developed the initial algorithm. Aishi contributed. And then I want to give a shout out to Erwin Perez, who's a summer intern in a BioInfo-Core, who helped contribute to the results as well. And here are my references.
Thank you very much. Thanks for a great talk and excellent timekeeping. Do we have any questions? Okay, let me just check. Are there any online questions? Where's the online bit? Oh, we have a question. We have a question.
Sorry. I wanted to, so you said you needed a certain amount of samples in order to this to work, and that it wouldn't work with low samples. What would you say would be the minimum number of samples for it to be worth using this method?
I would say that with 3 subjects, you could use the method, but the error bars would probably be overlapping for a lot of the resolutions. So it might give you like a general idea of what range of resolutions would be appropriate, but I think you ideally at least 6, but we haven't benchmarked specifically like what the minimum number is. So.
Okay, thank you.
Okay.